All right. Hello, everyone. Welcome to the first Science of Alt Protein APAC Edition seminar of 2023. My name is Manasa, the SciTech specialist for cultivated meat at GFI APAC, and I will be your host for today. It gives me great pleasure to welcome Dr. Shigeki Sugii as our speaker to talk to us about redefining adipocytes for cellular agriculture of alternative fat. Dr. Shigeki leads the Laboratory of Fat Metabolism and Stem Cells under the Division of Cell Biology and Therapies at IMCB A*. He also holds a joint appointment at Duke-NUS Medical School. Dr. Shigeki received his PhD in biochemistry at Dartmouth School of Medicine in the US, and then conducted his postdoctoral research with Professor Ronald Evans at the Salk Institute for Biological Studies and Howard Hughes Medical Institute, USA. Dr. Shigeki was a co-founder of Cellular Private Limited and recently founded Impact Fat in Singapore. Before I hand over to Dr. Shigeki, I would like to make a few quick announcements. We will be hosting another webinar in a week's time to highlight the use of mung beans as a key ingredient in plant-based foods. This webinar will be co-hosted by my colleagues, Dr. Wasamon Nutakul and Jennifer Morton, and we would love to see you all there on Thursday, 16th March. And the registration link for this will be provided in the chat window. Also, applications for the Alt Protein project are now live. The Alt Protein project, or APP for short, is a student-powered movement to seed alternative protein hubs at universities around the globe. Through the APP, student groups work closely with GFI and a global community of peers to build alternative protein courses, research projects, and startups at their university. Applications are due by 11:59 p.m. GMT on March 31st. If you have any questions or would like to know more about this project, please visit our website or reach out to us using the links provided in the chat again. Today's seminar will run for a total of an hour with a 15-minute slot towards the end for Q&A. On the bottom of the screen, you will see the chat and Q&A buttons. We encourage you to use the chat function to introduce yourself to other participants. However, please make sure to direct your questions to Dr. Shigeki in the Q&A window so that we can address them easily. In the Q&A section, you will also have the option to upvote questions you would like to be answered so that we can prioritize those that are most relevant to the audience today. For those of you who have registered for our speed networking session right after the seminar, please log out of Zoom after the Q&A session and log in to Meetaway using the check-in link emailed to you. We kindly request that you do not check in before 11 a.m. Singapore time and that you stay for the entire duration of the session to allow for effective attendee matching. Upon checking in on Meetaway, a 1-minute intro video will be played at the beginning, followed by a series of prompts to get you started. The session will run for a total of 30 minutes, where you will be matched with another attendee for a total of 5 rounds of 6 minutes each. We encourage you to keep your introductions short and sweet and to use this opportunity as a springboard to set up follow-up conversations and meetings. With that, Dr. Shigeki, please take it away.
Hi, everyone. I hope you can hear me okay and see my slides okay. Thanks, Manasa, for the kind introductions, and thanks GFI APAC to invite me to speak today. So I will talk to you about the cultivated fat as a potential for alternative fat. I focus on the fat cells, but I hope that many of the aspects are also common for other cell types like muscle cells. Starting with a disclosure, so I'm a co-founder of the Impact Fat, which was incorporated just 1 year ago. And some of my research I talked to you today may be related to commercialization effort of Impact Fat. So I think all of you know this, but we started to see more alternative protein products like plant-based meat products like Impossible Foods and many others. And we also started to see the product, cell-based meat product. This is a good meat product by eJust company. which was approved in Singapore for the first time in the world a few years ago. But of course, these products are good in the taste, but there is one still issue that many people feel and claim, that they lack something similar to the fat that gives you the taste in mouthfeel. And the best example of the contribution of fat is probably in the marbling of the beef, because many of the beef, especially those premium products like Wagyu beef, the more premium, they tend to have a higher amount of fat tissues or fat cells, and some reaching even higher than 50%. In the case of the fish, like sashimi, the best example is the tuna. So the so-called otoro, chutoro has a high amount of the fat cells that gives you some juiciness and premium as a products. And there is various studies studying the relationship between the fat amount in the meat and overall acceptance rate by the consumers. And this review compared and compiled all the studies. And so in general, there is a very good correlation between the fat amount and the probability of the acceptance, like tenderness, juiciness, flavor, and overall liking. And speaking of the fat as a food source, there are different kinds of the fat. Of course, traditionally, you have been eating like plant-based oil, like used in cooking oil and so on, or animal fat when you eat meat or sashimi or fish fillet. But there has been lots of development towards a novel fat, especially fat mimetics. So many researchers and companies are processing oil to make a fat mimetics, or some fermented lipids by using microbes. But our interest is using cultured cells or cultured fat by using the cellular agriculture techniques. So when you eat fat, actually from the meat products or fish fillet, what you eat as a fat is made of the fat cells mostly. And fat cells, of course, are the complex in structures, but they are really specialized in storing large amount of the fat molecule, which is lipid molecules, in the organelle called lipid droplets. So by that, they can stably store the large amount of the fat molecule inside the organelle. The fat cells may also contain bioactive proteins and other nutrients. But the typical structure is semi-solid, so that it doesn't go away during cooking, but it gives you juiciness when you eat. And theoretically speaking, you can potentially produce in an unlimited quantity. And but since there has been very little studies towards the production of the fat cells for the food products, more studies are necessary. But when we could make enough amount of the fat cells, we may need a minimum processing to incorporate into the meat products. This may be in contrast to the lipids, where the extensive physical or chemical processing is necessary to complement into the alternative protein products. So actually, fat cells, or we call adipocytes, are really well studied, but the vast majority of the studies are done in a human or rodent species like mouse or rats, mainly in the context of the obesity and metabolic diseases, which I was also working on fat cells in using human or mouse for a longer time. And this shows the number of the publication in the PubMed, that's the database in the top, or Web of Science in the bottom. So that there are thousands of papers published each year in using human or rodents. There is some limited number of the studies, but there are some studies using the cells from cow, pig, chicken, fish, which are related to the food. So we decided to look at those papers, especially those adipocyte-related papers, using the protocol to show any of the aspects shown here. So for example, the fat tissues are isolated from animals and cells are also isolated from tissues by enzyme. And then afterwards, the cells are centrifugated, pelleted as a stroma vascular cells. This contains a stem cell population or some pre-adipocyte populations and can be separated from mature fat cells, which do not grow in culture. So basically, people grow the SPCs to enrich either stem cells or pre-adipocyte populations. And then after growth, then differentiate it back into mature fat cells. And there are various technology protocol to analyze the adipocyte, like analyzing by the staining with a dye, or analyzing gene expression by qPCR, or analyzing the protein by Western blot, and so on. So first, we decided to look into those papers which describe the protocol related to adipocyte culture from bovine, porcine, chicken, other livestock, or seafood like fish species. And the total is over 500 papers. And this just shows an example of the table we compiled. And I won't go into detail because it was already published recently. So you are encouraged to get the paper and read the table if you're interested. But basically, we compile those title of the papers, what tissue source they isolated cells from, what the isolation methods, cell types used to grow, or growth conditions for growth or differentiations, or adipogenic genes or proteins that were investigated, and analysis methods for adipocytes, and finally link to the reference. And this also shows an example of the graph we compiled. So as for the isolation methods, almost all the paper use an enzyme called collagenase, but the different types, mainly like type 1 or type 4. And the cell types described, the main cell types are stroma vascular cells, which is actually the mixture of the different cell types. But many papers just describe as a pre-adipocytes, but from different sources like subcutaneous, intramuscular, or visceral fat. And the basal media they use to grow the cells are usually divided into either DMEM or DMEM-F12 mixture. But there are also some basal media found to grow the cells. Serum, as for serum, almost all the papers use the FPS, filtered bovine serum, but somewhat different concentration, but 10% is the majority. As for differentiation media, also the basal media is similar, DMEM or DMEM-F12. As for serum, also FPS is very common, but more variety in terms of the percentage, or some paper use other serum from other species. As for differentiation cocktail, so you need some stimuli to differentiate from stem cell or pre-adipocytes into mature fat cells. And the three major components used is dexamethasone, insulin, or IBMX that is used to increase cyclic AMP. Interestingly, some species like chicken, the fatty acid, oleic acid is commonly used to stimulate adipocyte differentiation too. This just shows a scheme of the adipocyte differentiation. Actually, adipocyte differentiation or adipogenesis is a quite complex, the process involving many different pathways. But basically, after stem cells are committed to become pre-adipocytes, there are divided into 2 or more stages: early stage adipogenesis and late stage adipogenesis. And accordingly, typically adipocyte differentiation cocktail also need at least 2 stages. And we also compile in a table what components are used for the first early stage adipogenesis versus late stage adipogenesis. As for genes, so many paper look at those adipocyte-specific genes like PP gamma, AP2, also called FABP4, CBP alpha, lipoprotein lipase, or fatty acid synthase, and so on. And similar for the proteins, the same proteins factors are investigated. As for analysis methods, the most common is staining with Oredo, which is easiest. But many people also look at the gene expression like by qPCR or protein expression by Western blot. So now I'd like to talk about our efforts toward making the cultivated fat from the fish species. And why fish? Because we believe that it has a better consumer perception than the livestock fat. And it is known that the fish fat usually have a less saturated fat and known to have a relatively high amount of unsaturated fat, including omega-3 fatty acids. And it may also offer good organoleptic properties. And again, showing a similar diagram, but this is our scheme for producing cultivated fat. So after we isolate the cell, we establish stem cell lines, and then we need to establish the growth conditions, first in a 2D, but eventually as a 3D conditions. And then we also need to find the adipocyte differentiation conditions. And afterwards, we can mix together the adipocytes with muscle cells and edible scaffold so that we can make cultivated meat or cultivated fillet products. And our work was supported by the funding from Singapore Food Story R&D program. And as for the fat as a nutritional sources, it is recommended that up to 30% of the fat intake is recommended because fat is actually one of the major 3 macromolecules along with the carbohydrate and proteins. But not all the fat species are created equal. And I'm talking about the lipids here, but the so-called unsaturated fat is considered good, but the saturated fat or trans fat is in general not so good. So the aim is to increase the good fat like monounsaturated or polyunsaturated fat like omega-3 fatty acids. And fat cells are also specialized in storing large amount of fat-soluble vitamins like vitamin A, D, E, K. So the first we did was to isolate the cell lines from edible fish species. And so far we have established cell lines from the patin catfish, is also called the pangasius catfish, or barramundi or sea bass, Japanese eel, also called unagi, and Australian jade perch. And this is an example of the pangasius-derived cell lines. So we typically screen through the hundreds or thousands of the single cell clones. And there are some variations in terms of the growth characteristics, but we typically pick up the best growth characteristic ones. And when we compare those fast-growing cell clones versus slow-growing cell clone, we see significant differences in metabolism, like fast-growing cell line consume more glucose and other amino acids like glutamine and so on, compared to the slow-growing cells. And we pick up, establish the cell line from pangasius catfish. We found that they have a very good growth characteristics, typically ranging from 13 hours to 24 hours, depending on the clones. But yeah, this particular clones grows as fast as 13 hours. And another characteristics is they can continue to grow and does not really undergo senescence in the right culture conditions. So this is without any genetic modifications. And so far, yeah, they grow more than 170 passages. Actually, yeah, more recently, it reaches almost 200 passages that translate to the 400 population doublings. So the standard culture conditions, yeah, we still need serum as the best culture conditions. And we found that 15% to be the best for this particular pangasius cell lines. And in addition, we also found that a small amount of fish serum is important in the optimum growth of our cell lines. But yeah, we found that 0.1 or even 0.05% to be optimal. But of course, yeah, there has been lots of effort. We trying to stay away from the serum. So this shows the growth curve of our standard media condition DMEM plus 15% FPS. But we tested the various commercially available serum-free media that are optimized against the human cells. But some seems to work to some extent, but still need a fish serum for the long-term growth. So speaking of the passage number, so we compare those young passage number cells versus old passage number cells. And we actually found that the higher passage number, they tend to grow even faster without a loss in differentiation capacity. And we also try to reduce the amount of FPS, and we found the customized media compositions. In this case, we found that we could reduce to as low as a 2% FPS. Although, we still need a small amount of fish serum, and the basic FGF growth factor is even more important in the serum-free condition or low serum conditions. And obviously, this is not a sustainable way of making fat. So definitely we will try hard towards serum-free culture conditions. And for that, this is the example of the collaboration we do with the CIFP. It's another institute in A-Star that's focused on the food. So this particular group has a number of the plant-based or fungal-based extracts, the library. So together with them, we screen through the thousands of the extracts. And after 3 stages of screening, we have some good candidates, 10 plant-based extracts or 2 fungal extracts. So hopefully, we are right now doing more testing and different conditions, and we hope to identify those or combination of those which can replace the serum. So next is the conditions for differentiations. And for that, we first tested the typical cocktail used for human or mammalian cells that consists of dexamethasone, insulin, IBMX, together with a PPA gamma agonist. But this does not really work for our fish cell lines. This is a fluorescent staining. So the blue is a nuclear staining, and the green is a Adipo-red staining that stains the lipid droplets. But we managed to find a condition that induced the differentiations of our fish cells. And we found that the 6 days is sufficient to induce the differentiations. And surprisingly, they exhibit almost close to 100% efficiency in adipogenesis, which we have never achieved with the human or mammalian cells before. We also tried to use serum-free conditions for differentiations. So as for differentiation, it was possible to use the serum-free conditions and actually even better in terms of the total lipid intensity. And again, it takes 6 days to become fully mature adipocytes. And adipogenesis efficiency is also close to 100%. This is just showing how we analyze our adipocytes. So basically, we use image-based analysis method, which we described a couple years ago. So we count the numbers of nuclei and then identify the lipid droplets in the vicinity of the nuclei, and the measures percentage of the adipocytes or nuclei that has the lipid droplets in its vicinities. We also do gene expression analysis of the adipogenic genes like PPA gamma, CBP, beta, alpha, hormone-sensitive lipase, so lipoprotein lipase, and fatty acid synthase. And unfortunately for fish cells, it is quite challenging to do the protein analysis because there has been really no antibody that works well against the fish adipocytes. So if you happen to generate any good antibody, or you are interested in generating good antibody against fish proteins, we really love to work with you. The same or similar adipocyte differentiation condition also work for other fish species, cell lines, such as Australian jade perch or Japanese eel. And yeah, it also takes actually between 3 days and 9 days. Australian jade perch takes 9 days to differentiate. Japanese eel actually takes only 3 days to differentiate. So one of the concerns that we heard is, since our cells grow so well, is it due to the activation of the cancer-related genes so that cells may be cancerous? So to address these questions, we did the whole transcriptome analysis by using RNA sequencing. And we basically compare our cultivated adipocytes with the original fat tissues taken from the Pangasius, and did gene ontology analysis and found that more than 88% of the genes expressed by our cultivated adipocytes are also expressed by the original fat. And the only differences are mainly found in a different category by gene ontology, like immune response or extracellular regions. And we believe that this is due to the presence of other cell types in the fat tissues, because fat tissues not only contain the mature fat cells, but they also contain immune cells and some blood vessel cells along with the stem cell populations. Importantly, what we found is no tumor-associated genes are activated in our cultivated adipocytes. So yeah, this may clear one of the food safety concerns. As for nutritional profile, we did a metabolomic analysis, and we found that the DHA and EPA are stimulated and induced by adipocyte differentiation in our cultivated adipocytes. And we did a basic nutritional profile. This is still a preliminary results, so it's not really the final, but our preliminary results indicate it's actually fat cells contain quite a bit of the proteins, but also quite a bit of fat, and almost all of the fat found were unsaturated fat, and very little saturated and no trans fat. So finally, to make these protocols scalable to bioreactor-based production system, it is mandatory to establish 3D culture conditions. And as part of the effort, we just show you the one example using the microcarrier. So this is a commercially available microcarriers, Cytodex-1. And yeah, it is possible to grow on top of these microcarriers. And this is just blue indicating the nucleus of our Pangasius cells. And the same condition was used to induce differentiation, and that worked. So the greens now shows a lipid staining after adipocyte differentiation in a 3D culture conditions. So as a summary, we also established the cell lines from at least 4 species of fish. And fastest cell line exhibit the 13-hour doubling time and more than 340 population doublings. And we develop culture media and conditions for growing our fish cell lines. We develop a method to differentiate our cells into mature adipocytes. And these adipocytes are enriched in the unsaturated fat, including omega-3 fatty acids. And we develop the methods, scalable methods for in 3D culture conditions. And along the way, it is critical to establish the scale-up production system using bioreactors. And that is part of the reason why we spun off the impact fat. And as an impact fat, one of the focus is to translate this scalable protocol into the bioreactor-based production system. And yeah, recently, a few months ago, Impact Fat held the world first tasting event of the cultivated fish fat. And definitely once we can produce enough that impact fat can optimize the organoleptic properties of the cultivated fat, and investigating what kind of the products the cultivated fish fat can complement. So what I have been talking so far, so this is a diagram we recently published in a review paper. So for those interested, please feel free to read the reviews. But what we have been talked to you was in the left. So most of the protocol related to adipocyte culture, the current standard. But we still need to develop more, and we need much more options and better technologies to develop this as a whole field. So, for example, we still need more cell lines that are hopefully the good characteristic of the growth and differentiations. We also need to establish bioreactor-based system. And what kind of bioreactor is also more investigations are necessary. We also need to need more scaffold material, and hopefully it will be edible so that we can eat as a whole. Or we could develop the methods for spheroid or organoid culture. We also need better characterization methods like live cell imaging, or we need to establish more cell surface markers that can dictate the cell types or quality of the cells. And we also need critical quality attributes so that we can monitor the productions and making sure the cells we produce are the good ones. We also need to establish more organoleptic and nutritional profiles. And finally, when we incorporate our fat into the food, how we can do it is still lots of studies necessary. And for unstructured meat, it may be relatively simple, but what kind of scaffold, material or cells to mix, we still need some studies. For structured meat products, definitely need further studies, research, for example, using 3D bioprinters. So this, obviously we cannot do it alone, and we hope to work with many of you towards many of these aspects listed here. So finally, I'd like to acknowledge all the people who contributed to the study I talked to you today, including our members, especially Lamani, Cheryl, and other students in our group, and our collaborator from Temasek Polytechnic, Republic Polytechnic, SHIFB or NTU Fresh, and finally, we'd like to acknowledge the member of the CRISMIS program, where we are involved as a national project towards cultivated meat development. And like to acknowledge the funding support as listed here as well. So I think I finished a little bit early, but if you have questions, you may have enough time to answer. So thank you for attention.
Thank you, Dr. Shigeki. That was a really great presentation, lots of exciting work. And I myself have a long list of questions to know more, but I'll first defer to the Q&A window where some of our attendees have keyed in their questions already. For starters, a question from Claire. Thanks for a wonderful presentation. Great work on these cell lines. Are you open to sharing the lines your team has developed with other researchers in the field of cellular agriculture?
Sure. That is why we are part of the CRISMIS program. And one of the objectives of the CRISMIS program is to provide the cell line we developed. So CRISMIS has the dedicated team of the biobanking so that they can do the all the quality assurance work. And we have for example, we have a Pangasius cell lines, and they did all the quality assurance work. So this is now ready for sharing. So please feel to contact me or contact the person in charge of the CRISMIS if you are interested.
Great. There is another question from an anonymous attendee who says, for the scale-up protocol, do you plan to use Cytodex-1 in the future? I wasn't sure if it was edible. If not, what kind of scaffold or microcarrier are you planning to use?
Yes, yes. So the Cytodex-1, as you said, is not really edible. It's a dextran-based, so it's not the, it's not harmful, but it's not edible. So definitely we are looking for the edible scaffolds, and yeah, several collaborations are ongoing. But if any of you has a good edible scaffold, we are also open to try with our fish cells.
That's great. There's a question from Ian who asks, have you tried adapting to single cell suspension?
Yes, that's a good question. We also tried spheroid culture, but we have some challenge with our fish cells because our fish cells have a relatively weak in adherence. So it can grow to a certain size, but relatively small spheroid size, and then they stop growing. So unlike some of the human, fat-derived stem cells, so far there is some limitation in a spheroid culture. So, and the single cells, actually, they cannot grow in single cell. They need to actually grow in touching, right? The communicating to each other. So it can grow in a spheroid to some extent, but it cannot grow in a single cells.
Great, thank you. Actually, I had a question on the growth aspect of the lines that you presented as well. It's really, really exciting to see the short doubling times and the number of passages that the lines are capable of growing to. So I was just wondering, in your comparison of the different cell lines, did you see that the fast growers were also the best ones to differentiate?
It's not necessarily the case, but yeah, we typically compare the different kinds of the cell lines with the different growth characteristics. And in general, there is a rough correlation between the growth, faster the growth, the better the differentiations, because one of the characteristics of the adipocyte differentiation, they need to become more confluent. So, more dense, they tend to differentiate better. So the faster growing cell lines has some advantage. But I don't think, biologically speaking, the fast growth does not necessarily correlate to better differentiation.
Got it. And in terms of the whole genome sequencing analysis that you did, there's a question here from Adam that says, would it matter if there was upregulations of tumor-associated genes from a scientific perspective? Would there actually be any safety risks to the consumer or otherwise?
Yes, yes. So that is definitely being debated. So theoretically speaking, right, eating cancerous cells, may not be really the risk, I would say. But of course, consumer acceptance is a different story. And accordingly, regulatory bodies usually look at this carefully. So it is best that the cancer cells are avoided, or any cancer-related characteristics are avoided when developing food. But I wouldn't say it's not acceptable at all when it comes to the food products. But you'll be scrutinized carefully.
And along the same lines, I guess there's a question as well, which asks, are your fish adipocytes spontaneously immortalized?
That's a good question. Yeah, we don't know really the answer yet. But I would say, yeah, we can safely say it may be spontaneously immortalized, or possibly it may already acquire the immortalizing characteristics. But yeah, we don't know the good answer yet.
Okay. And there's a comment.
Oh, by the way, yeah, so when talking about immortalization, right, the telomerase needs to be upregulated. But
right.
So far, we don't really see the big upregulation of the telomerase. So we may not call it spontaneous immortalization.
Right. Okay. Yeah, that's interesting because it just may be that they have a very long replicative lifespan before they eventually senesce.
Right.
On the same lines, did you notice that the number of population doublings that you were talking about, was that specific only for the catfish, or was that seen across the other species that you tested as well?
So you're talking about RNA or gene expression analysis.
I'm talking more about the growth profiles. For
Growth
example,
profile. Yes,
yeah.
yes. Yeah, we have done the growth characteristics. So typically the doubling time is between 15 hours and 24 hours. It varies from cell line to cell line, but in general, yeah, they exhibit less than 24 hours.
Okay, excellent. There's a comment from Jian Hong that says, great talk, not really question, but for fish proteins without antibodies, you could try to use mass spec to quantify. I'm not sure if that's something you've considered already.
Oh, yes, yes. Yeah, I think he is a specialist in mass spec. So yeah, if you have a good capability of mass spec, you could do it. I wish that we could do it routinely, but it's a little bit more challenging to quantify with mass spec. But when we have good products, then yeah, we are also keen to measure all the proteomics analysis.
Yeah,
and actually, yeah. So Impact Fat is doing the safety analysis. Part of it is allergen analysis, right? Allergenicity analysis. So the proteomics is actually used as part of the allergenicity analysis.
All right. Then there's a question from an anonymous attendee, which is it necessary to enrich the adipose stem or progenitor cell population? I'm not really sure whether that's within the context of differentiation that they're asking, but have you in your studies felt that during your isolation procedure, there's a specific step that you do to enrich for a certain population?
Yeah, well, I wouldn't say the specific cell line per se, but it is important to establish the good cell lines which can grow well and differentiate well into mature cell types. So it doesn't have to be stem cell population or pre-adipocyte population. And by the way, so far we cannot really define the specific cell types because there's really no good cell surface markers for our fish cells. But as long as you get a good so-called progenitor cells that has a good growth and differentiation characteristics, it may be sufficient for now.
Okay. As a question from Danai related to the media that you used, most of the groups working with fish cell lines are using L15 media. However, I noticed that you found DMEM to be superior. In which trait specifically are you referring to? Is it cell yield or cell doubling?
That's right. So we have used L15 as alternative media because L15 has been traditionally used often for the fish cell culture. But what we found is still DMEM is better than L15. And if you look at the compositions, DMEM has much more nutrients, and that may be why. But in certain situation, the L15, because certain situation, you don't want to use CO2 incubator, then L15 media is also possible to use it.
Actually, on the topic of media, I was also wondering on the collaboration that you're working with SIFPI on for the serum replacement extracts. Have you identified particular types of or classes of molecules that you actually want to replace? Like, for example, is it broadly growth factors or lipids, or is it more generic than that?
Yeah, good question. I wish I could answer your questions, okay. But we need to work harder to identify what kind of the molecule is it, or even protein or some other components. Unfortunately, so far we don't have a good answer, but we plan to try to identify as much as possible what would be the factors. But having said that, still, certain growth factor is important, especially basic FGF. So these factors may not totally replace the FGF.
Yeah, okay. And there are two questions which are, I guess, relating to the organoleptic properties of the different fish fats. the attendees want to know, are the different cultured versions different in the organoleptic properties? And can you actually taste any difference between the different cultured versions? I'm guessing from the different cell lines.
Yes, that is also interesting questions. We don't have a definite answer yet. So far, it is possible to taste internally. So we have actually tasted some the pangasius derived cells, but we haven't tasted other species. So yeah, we cannot comment on the differences in the taste. But looking at the fat components of different fat, different fish fillet, you may see some differences in the taste, right, and texture. So I expect that there may be differences. And as you may see in our adipocyte culture, so for example, our adipocytes from the unagi cell line, they somewhat show the different morphologies. So this may lead to different textures and potentially taste. But having said that, I think taste is customizable by using different components in the culture media. So yeah, I don't think I have a good answer for your questions, but yeah, that is the best I can.
Okay. And there's also a question just regarding the general approach that you've taken for culturing the adipocytes. Adam asks, as far as my understanding goes, animal lipids can be created through adipocyte isolation, differentiation, etc., or through fermentation, that is directly making the lipids and isolating the lipids themselves before incorporating into the food. Can you talk a little bit about what led you to go down the former route and not the latter? And are there any advantages or drawbacks to either of these two approaches?
Yes. So the questions, I know that there are many people working towards, producing lipids or using lipids to complement alternative protein products. So there are pros and cons. I think the major, of course, you can make in a bulk and in a cost-effective manner, but the potential problem is that melting temperature, for example, they tend to be more dependent on the lipids you use, so that during cooking it may become liquid and go away. Or it's also more susceptible to chemical changes like oxidations. And then it can get rancid, and as a nutrition, it's not good anymore.
Okay.
So yeah, on the other hand, we believe that if we could make fast cells, this is something really identical or at least similar to what you eat in the animal meat and fat components. So it's relatively easy to recapitulate all the organoleptic properties of animal fat.
Right. And I was also wondering, you spoke a little bit about how incorporating the cultivated fat is going to be challenging for more structured meats as well, right? So do you see like fermentation-derived lipids actually being easier to incorporate in terms of practicality than the adipocyte-derived?
Mm-hmm. If you're talking about using this microbe, right, which contains the lipids, it is, of course, theoretically possible and interesting techniques. But I don't know if it gives the same textures, because microbes typically has a large cell wall. So don't know if we can give you the same juiciness as we found. And also microbes is not really naturally specialized in storing large amount of the lipids. So there may be some limitations in the storage of the large amount without any chemical or physical changes occurring. Okay,
great. We have time for a last few questions. There are a couple of related questions around just the general status of your cell lines being qualified as non-GMO. a couple of attendees want to know, is this based on the Sendai system that they are non-GMO, or has it been qualified as non-GMO? And do you see a reduction of P53 or any other cell cycle checkpoints in the gene expression profile?
Yeah, so as for the non-GMO, we believe that it's considered as a non-GMO because we didn't use any genetic modification. So we didn't introduce any genes. So no system was incorporated in generating our cell lines. As for the second question, P53 cell cycle, I don't recall out of my head right now, but P53, if I'm not wrong, there was no change in the gene expressions, and cell cycle genes also not so many genes that came up. At least, yeah, I don't think there is any upregulation of the cell cycle related genes.
Okay, great.
But I may be wrong.
Okay. There's a final couple of questions. Again, just going back to the part where you spoke about holding a tasting session. There was an attendee who's curious to know, how did you manage to hold the tasting session when using dexamethasone? And are you planning to move away from standard differentiation media?
Yes, so dexamethasone, yeah, as you may know, is a pharmaceutical drug. And in the cocktail, right, there may be some molecule used, which was evaluated by SFA for the safety. So what is really important is potential final, the estimated concentration of these components remaining in the cultivated fast cells. And fortunately, all these components we use in the first stage of adipocyte differentiations and not the later stage of adipocyte differentiation. So, and afterwards we have a washing steps. So the estimated conditions, concentrations, yeah, that we calculated is well below the acceptable limit as a food product.
That's good. Thank you for highlighting that, Dr. Shigeki, because I think that's a common question that a lot of people have. And there's also a question from Apeksha here related to that, which says, in one slide, you showed that day 6 was optimal for differentiation. Was the adipogenic cocktail added on days 3, 4, 5, and 6, and then left until day 9 and added again? So if you could please just elaborate a little bit more about like the steps that you followed and at what stages the differentiation was actually induced with the cocktail.
Yeah, so we have 2 stages of differentiations. Some are proprietary, but we basically use some of the traditional cocktail plus the mixture of the lipids and in different stages. So that is, yeah, used to make 2 stages differentiations.
All right. The last couple of questions here. Chuan Lee is curious to know, you mentioned that the cells might have obtained spontaneously immortalized characteristics. Is this purely through subcultivation? Or was any other method used?
Right. Yeah, I don't think I have a good answer to these questions. But we basically screen through the single cell clone. So we naturally, selected those which, exhibit the more like immortalizing characteristics. So I don't know if that's what you mean by subculture, but we did some high throughput screening to identify the best cell line.
Okay, yeah, I think that's what the question was referring to, as opposed to using any sort of GM or GE techniques. It was through single cell cloning and subculturing. All right, and then David in the chat section has also wanted to know whether this work on your cell lines has been published already in a journal.
Oh yeah, we are working on paper, and we are trying to publish the results we have shared so far. And we also describe in a PCT patent all these. Okay,
excellent. We look forward to the publication. And finally, Dr. Sugii, I think a point that you highlighted very well is that there's a definite absence of suitable antibodies that work well against fish cell lines and that we need more work to be focusing on developing these antibodies. So I just wanted a little bit more information from you as to what are the biggest challenges are for developing these sorts of fish cell lines. Is it solely now the bottleneck being characterization, or do you see this in addition to other white spaces that need to be addressed?
Yeah, so there's no single good answer, I would say, but definitely characterization is one, because there has been, very little studies done in many of the fish species, and we have so many fish species.
Yeah.
Main fish work has been done in a zebrafish, right, and some in a fugu or limited species. But even those, very little studies on the adipocytes. So definitely we need more understanding of the basic biology of the fat and adipocyte biology. Which is still missing, and accordingly, there is also lack of the markers and antibodies. These are also related to how best we can isolate the cell line, right? So not only the fat cells, this applies to muscle cells and other cell types. So yeah, I don't know if I could answer, but and also, yeah, for scale-up productions, it may be different from many human or mammalian cells, which has a longer history of the scale-up production. So that could be also another challenge.
Okay, so bioreactor design and production processes may also need tweaking away from traditional processes that have been used for mammalian cell lines as well, I guess.
That's right, right. So in our experience, many of the protocols are not identical when it comes to the fish cells. So we actually need to optimize or develop the new protocols.
All right. With that, we just have a couple of minutes left to finish. So if you have any pressing questions to ask Dr. Shigeki, please do so now. If not, you may reach him via email. I've just posted his email address in the webinar chat. But thank you so much, Dr. Shigeki. This was a really good session, and it's been really insightful to see all the work that is being done at Impact Fat and what needs to be done. And I really urge the attendees to please get in touch with Dr. Shigeki if you want to collaborate, if you would like to know more about his cell lines to see how you can use it for your work. But apart from that, thank you again. And for the attendees who have registered for the networking session, we will see you at 11 in 3 minutes on Meetaway. But thank you very much, Dr. Shigeki.
Thank you, Manasa, and thank you all.
Thank you.